Each cell in the human body contains about 2 meters of DNA that's packed into a tiny nucleus that's only about 5 micrometers in diameter. Transcription occurs in the nucleus of eukaryotes. It involves the creation of RNA from the genes in the DNA. Certain proteins floating around in the cytoplasm occasionally travel to the nucleus when it senses a need for transcription of certain proteins. This signals for the activator protein, the mediator protein, and the tatabox to come together and bind with the RNA polymerase at the promoter site. This initiates the start of transcription and opens the double-stranded DNA to expose the DNA template strand. From there, the RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA. When a eukaryotic gene is transcribed in the nucleus, the primary transcript isn't yet considered a messenger RNA. Instead, it is considered an immature molecule called a pre-mRNA. The pre-mRNA has to go through some modifications to become a mature mRNA molecule that can leave the nucleus and be translated. These modifications include splicing, capping, and addition of a poly-A tail. The mRNA is first spliced using an enzyme called a spliceosome, which is composed of several small molecules called snRNPs, or SNRPs. This process removes the introns, which are segments that will not be expressed, and combines the exons, which are segments that will be expressed. Next, it is capped, which adds a 5' cap to the 5' end to prevent it from being digested by nucleases and promote translation. Lastly, the mRNA undergoes tailing, which adds a poly-A tail to the 3' end and protects it from termination of translation. Finally, after the premature mRNA turns into a mature mRNA, it travels to the nuclear pores to prepare for translation.